Welcome to another episode of the Dendrite podcast with Said and Nika. This is a conversation with Dr. Darren Blackburn. He is a postdoctoral fellow in Dr. Soleimani's lab in the Department of Biochemistry, Microbiology and Immunology, Faculty of Medicine at University of Ottawa.
During his scientific career, Dr. Blackburn has investigated the molecular mechanisms underlying cancer cachexia and their effects on muscle stem cell function. He has previously worked on ecology, entomology, and parasitology of different weeds and bug species. We hope you enjoyed this episode, and here's Dr. Darren Blackburn. All right, good morning. This is a really beautiful morning after a while. Like, I just got outside and I was like, I just want to walk. I didn't at the end of the day, but would be nice maybe on the way
weather's
back
getting
home.
warmer.
Yeah. So, thank you very much for accepting our invitation. Would you mind tell us a bit about your journey?
Sure. I guess my academic journey is described as, I suppose, meandering. I started, my first degree was a diploma in biotechnology at Algonquin College. It's a good program. I enjoyed it. It taught me a lot on lab techniques, molecular techniques. And then afterwards, I got my bachelor's in biology at Carleton University. And there, I did not work in biotechnology at all. I worked more in ecology, entomology. Parasitology was probably the major thing I was working with. So a lot of my research during my undergrad and a bit after was on biological control. So invasive species and how we can control their spread using their natural predators. So, for example, since the world is getting more global, has nothing to do with translational medicine or anything, but more and more species are able to travel across the globe and are able to integrate into environments that they're not native in. And to be an invasive species, you have to be a nuisance. And we describe that as an economic nuisance. So, for example, earthworms are not considered an invasive species because they're not viewed as a nuisance. But, the killer hornets in Vancouver, that would be considered an invasive species. So I worked a lot in parasitology. So that's the main thing on controlling invasive species is we find their natural predators. Parasites are a good one, working mostly with parasitoids, which is a slight variation on it. So during the undergrad, I worked with invasive weed species, so plants that were coming in, and looking at their parasitoids and seeing looking at their biological control agents, which were not parasitoids, they were bugs, and seeing whether their natural predators also came along with them and were in fact controlling the biological control agents themselves. And the answer was yes, some of the parasitoids for those had come along as well, so they're now present for those biological control agents. And then I moved on to arthropod biological control, so working mostly with an invasive stink bug species. Maybe you've heard of it. It's the brown marmorated stink bug, the Haliomorpha halis. It is in Canada, but we don't really care that much on it. It's more of an issue because one of its favorite foods is hazelnuts. So Turkey is very concerned with it. Italy as well. They're major producers of hazelnuts. They eat other things.
Their taste and
Well, they kind of destroy the hazelnuts, so it doesn't really help their crop yields. They do eat beans, carrots, corn, they eat a lot of other crops, but it's mostly hazelnuts. And we were working on whether an egg parasitoid could be a viable biological control agent. And what I was working on, whether this wasp, it was a parasitic wasp, would be able to target these Haliomorpha halis eggs, but not target the native stink bug species, because you didn't want it to target the native species, you wanted to only target your agent. And then afterwards, I was like, I want to try something else, why not? So I applied to McGill for a master's in human genetics, and I didn't choose muscle stem cell biology or regenerative medicine or anything. It just kind of randomly happened. I was applying to labs. Most of them were interested, but they were like, we don't have funding for a student right now. You should go try Vahab Soleimani's lab. He works with muscle stem cells. I was like, sure, I'll apply to his lab. And coincidentally, maybe this is how life is odd, but my mentor in Carleton Dr. Ian McKinnell had done his postdoc with Vahab. So when I told him, like, oh, I have an interview with this Dr. Soleimani, he's like, oh, I did my postdoc with him, great guy, like, very good scientist. And I only learned this after I joined Vahab's lab, but we also worked at the same institute. So when I was doing my undergrad, I worked at the experimental farm. Vahab had also worked at the experimental farm. We had actually been in the same physical lab, not the same scientist, but the same room. I learned that after, because when I was at the farm, people would tell me, oh, the previous researcher in this room, here's all the stuff that happened. And then Vaha would tell me stories of when he was at the experimental farm. It's like, I know these stories. The same stories I was told in our room, and we figured out we had the same space. So it's a weird overlap and coincidence in our backgrounds. And anyway, so when I started my master's, I knew nothing on muscle stem cells, muscle stem cell biology, but I did enjoy it a lot. So I fast-tracked to my PhD afterwards and completed that in 2023. And then I started my postdoc here at U Ottawa, working in the same field, muscle stem cell biology, regenerative medicine, and so on.
All right. And how does your background in ecology has helped you in the muscle field?
So that's interesting. I find it helps a lot because ecology is all about like the connections between the plant species, the animal species, the environment, the weather, and in the human body, you also have those connections between like the niche cells and how they interact with the muscle stem cells, and you do get trained more in trying to find those connections and how they affect the muscle stem cell function.
Interesting.
Yeah, it's very interesting because all of these are complex systems into one degree or another. And what I'm studying right now is more on the brain side, and it's on a macro scale. So just getting the different like MEG signals of the brain and see how this signal, if there are any dynamic like spatiotemporal signatures of like say this disease or the other. And then looking at it, you can not design, but you can look at a sort of a homeostatic environment of this different signatures. So some of them can be pushed to extremes, and then when you have different disorders. But then it's very interesting to look at how a cell is just living its life. That's also very similar. So it's this environment that you have certain variables that should behave a certain way so that like the whole system can survive. And now you can develop that into different organs, into different a living being, a society. So now when you say the things that you can translate or like the subjects or just the general information that helps you from ecology to muscle stem cells, this is very interesting to me because on face value, they're completely different.
Yeah.
Different life forms. There's a lot of differences. If you want to pick the differences, there's a lot. But similarities are a bit more subtle, but also very important.
Yeah, I find anything you learn, you can try to translate it and apply it to different contexts. And I find my background in ecology, agricultural work, it kind of moves over quite well here.
And also, what was one turning point in your career that shaped your whole career, other than, of course, discovering Vahab's lab?
I guess that would be the main one, but well, a lot of our work in Vahab's lab is based on niche interactions and how the niche cells communicate and affect muscle stem cell function. Mm-hmm. And I find in the field we do tend to focus a lot on like macrophages, fibroadipogenic progenitor cells or FAP cells, but we often don't think of the myofiber itself. So the muscle fiber itself is itself also a niche cell. It is the cell that's in direct contact with the muscle stem cell, and people often just think of it as the functional unit of the muscle. It contracts, it's what generates the force, but from our work in the lab, we also see that we've firmly concluded that it's also an endocrine tissue. So the muscle itself produces a lot of growth factors, a lot of signaling molecules that assist not just with the rest of the body. So we do think a lot of like the muscle-brain connection. The muscle produces a lot of neurotrophic factors that can assist in brain development and brain health, but it also generates those growth factors and signaling molecules important for muscle stem cell function as well. So a lot of the work that I did during my master's and PhD was on developing tools to study those muscle fibers in isolation. Because before the development of our single myofiber RNA-seq and ATAC-seq, most work on muscle is bulk muscle. So you're getting the myofibers, and they honestly probably represent over 90% of the muscle, but you're still getting the FAPs, the macrophages, the endothelial cells, the muscle stem cells, the neurons, and all of those get mixed together, and it becomes difficult to extract out what changes are happening in the muscle fiber itself and how that could be influencing the muscle health and the muscle stem cell function. So a good example on it is, in our work, we identify two key, they're called myokines. So we like to add myo to everything in muscle. So they're cytokines produced by the muscle.
Yep.
So two key ones that we identify is VEGF-A. So as most people would be familiar, it's the vascular endothelial growth factor, and it's very important for endothelial growth and expansion and branching. And the myofiber is the major source of VEGF-A, not just in the muscle, but in the whole body as well. So if you remove or reduce the ability of the muscle fibers to produce VEGF-A, you get a whole body reduction of that cytokine. And we also identify for the first time that EGF, the epithelial growth factor, a very important growth factor, is produced by the muscle fibers. So it is itself also the major source of EGF in the body. So yeah, as a whole, that kind of, I like to try to always bridge the myofibers with the muscle stem cells. So I'd say our work on that has shaped a lot of my future research as well.
I see.
So can I take a question back? So niche cells, what exactly are niche cells? Like different?
Yeah, so let's say, well, first let's define what the niche is. So we view the niche as the environment that a cell lives in. So that includes not just the cell itself, but also the extracellular matrix, the signaling factors, the circulating factors, and also its neighboring cells. So the muscle stem cell has a defined physical niche. That's actually how it was discovered, is it exists adjacent, on top of the myofiber, basically attached to the sarcolemma, so the muscle cell membrane, and also underneath the basal lamina. So, the basement membrane. So it's kind of sandwiched between the myofiber and the ECM. And that's its physical niche. I guess if you wanted to think of it in an analogy, it's picture a house. The inner wall is the muscle fiber cell membrane, the outer wall is the basement membrane, and the muscle stem cell is in between the walls. Yeah. So it's not part of the myofiber, but it's not outside of it either.
Not also housed between, because I remember back in studying anatomy, how muscle bundles work. It's like cables. So you have little bundles and then they come together and then you house all of them. So where the stem cell is like outside of all of them together?
No, so each individual myofiber has their own muscle stem cell associated to it. So yes, the cable bundle is the right definition, but in between every cable are muscle stem cells, and it's attached to one of the myofibers.
Oh, okay.
So it's not attached to multiple myofibers, it is associated with a single one. And yes, so every muscle has its own number of muscle stem cells per myofiber, so it is not actually associated to length or the muscle size. So for example, the flexor digitorum brevis, it's the muscle that lets you kind of flex your toes at the bottom of your feet. It's a relatively small muscle, but it has a very high proportion of muscle stem cells. I'm not sure why, but it does. In contrast, you have the extensor digitorum longus, so what lets you lift your toes up, which is a much longer muscle, and that one has very few muscle stem cells in comparison to the flexor digitorum brevis. So that's not really associated with size. But back to the question on niche cells, yes, so you have your physical anatomical niche, but you also have your physiological niche. So all of the niche cells are cells that are in that environment and are known to support the function of a cell.
All right, now taking a step back. So out of all of the diseases of muscle, there are so many different ones. Which ones do you focus on? What are they, if you want to define them? And how do you go about researching on them?
Sure. So there are a lot of muscle diseases. So let's start, if you'll bear with me. defining, I guess, the three major ones. So you'll have muscular dystrophy, sarcopenia, and cachexia. And I like, and it's good advice for everyone, to understand the etymology of words to really see what they mean and be able to apply that to other words as well. So if you start with dystrophy, while you'd be forgiven for thinking it's affiliated with muscle because you have dystrophin, that's the muscle protein. We all know about muscular dystrophy. Dystrophy itself means wasting. So when we say muscular dystrophy, we mean muscle wasting. But we only mean it really in the sense of muscular dystrophy, in the sense of like Duchenne's muscular dystrophy or the other forms of muscular dystrophy, where it's due to a genetic mutation in the dystrophin gene, which makes your myofibers more fragile. Therefore, they break down more often, and you are stuck in this constant loop of regeneration. So Duchenne muscular dystrophy is the most severe case, and that one, thanks to palliative care, patients do live a bit longer, but typically they don't survive much longer past the age of 20. Then you have sarcopenia. So sarco, it means flesh, but we always mean it in the sense of muscle. So anything that has the prefix sarco is, I shouldn't say every, because maybe I'm missing one and I'm not aware of it, but we always mean muscle. So like I was saying before, like sarcolemma, that's the cell membrane of the muscle. Sarcoplasm is the cytoplasm of the myofiber. Sarcoplasmic reticulum, it's the endoplasmic reticulum of the myofiber. So sarco means muscle. And penia means a loss or a defect. So when we say sarcopenia, we basically mean muscle loss. But that doesn't actually tell you what it means. Sarcopenia is an age-related loss of muscle. And there's no real clear clinical definition on it. And that's kind of what makes it challenging to define sarcopenia clearly. It's just, we tend to mean sarcopenia in the disease context. So when you get older, some people have a severe and major loss of muscle. We would tend to mean that to be sarcopenia. However, some could define sarcopenia as just any age-related loss in muscle. So typically after the age of 40, you do start to have this either gradual loss of muscle or a difficulty in maintaining your muscle. You could define that as sarcopenia as well. We tend to meet in the disease context where it's that major loss of muscle. Then you have cachexia. That one, if you do want to break it down, it means a bad condition. So caco is bad, hexia is a condition, your body condition. And despite the fact that it has no connection to muscle in its word, in its etymology, that one is specifically about muscle. So when we say cachexia, we mean a rapid, unwanted loss of muscle mass. The most common form that you would be familiar with is cancer cachexia, so the rapid loss of muscle due to cancer. Same problem with sarcopenia. There's no real clear clinical definition of cachexia. It's just you have muscle loss. Arguably, if I need to give a number, we would say in humans, in a year you've lost 5% of your body mass, which is a high amount. But cachexia can also apply to any chronic disease. So if you have chronic kidney disease, you can also exhibit cachexia, where your muscles will start wasting. Diabetes, I think, can also cause it. COPD. These are all chronic conditions that can cause you to lose your muscle mass, and therefore that would be considered cachexia. In my research itself, I work mostly with cachexia, cancer cachexia. Well, that's my current thing that I'm working on, but we have worked on muscular dystrophy, and we work on sarcopenia as well, because we are an aging lab. We do like to research aging and how it affects muscle function. And so that's my main thing right now.
So the naming is mostly trying to define what it's coinciding with. So for example, and the follow-up question would be, okay, so sure, we probably don't know much about like where it's coming from, but then what are their differences or in terms of their characteristic? So for example, let's say if someone starts to suddenly, well, by suddenly I mean just abnormal loss of muscle at the age of, let's say, 25 or 30, then which one would be our lead? So for example, it's not aging related, maybe,
right.
Perhaps.
They would be too young to be considered sarcopenia, but it would only be considered cachexia if it was some sort of chronic disease or cancer, and it may not be considered muscular dystrophy if it doesn't have that dystrophin genetic mutation. So then it would be, my guess would be some kind of cachexia, most likely, but I would need to see the patient and know what's going on.
Yeah, that particularly might be just a parasite or something. It picks such a unique thing.
All right, how different cellular signals affect myogenesis in general and also influence their quiescence, proliferation, activation?
Right. So for those who are not aware, muscle stem cells typically in a healthy homeostatic environment exist in a quiescent state. So that's a state where your metabolism is very reduced, protein synthesis is down, gene transcription is quite low, translation is quite low. They do not proliferate, they stay in a undividing state, but it's not senescent. So they are able to return into the cell cycle and activate. So in muscle injury, something happens, an insult happens to the muscle, and signals occur that tell the muscle stem cell to activate. We don't know everything, so we don't know every signal that exists, but some of them are, well, let's go through the process. So when the muscle gets injured, it breaks down, and that does result in a release of calcium. So muscle has a lot of calcium in it, it releases a lot of calcium, so that calcium signaling could be one of the triggering factors to start the muscle stem cells to activate. The other thing that occurs is immune cells come in. So macrophages come in to clear up the debris, inflammation occurs, and that inflammation is also a signal to the muscle stem cells to activate and proliferate. And then you have, well, let's go back to macrophages since I started on those. For example, macrophages release a molecule, GDF3, and that molecule is known to inhibit Notch1, and Notch signaling is important in maintaining muscle stem cell quiescence. So when the macrophages come in, they can inhibit one of the quiescence markers of muscle stem cells, promoting their activation. Then once the cells are active, the muscle stem cells are activated, they differentiate to myoblasts, they proliferate, they expand, and then they either fuse together into a new myofiber, or they'll fuse to pre-existing myofibers to assist in their regeneration. And then once you're regenerating the muscle, you need to remodel that ECM, the extracellular matrix, to bring yourself back to homeostasis. And that is often done, like FAPs are a major contributor of the ECM and remodeling that ECM. So they probably play a major role in remodeling it and helping the muscle stem cells return to quiescence.
So physiologically, when they say if you want to grow your muscle, you have to tear your muscle, that is technically kind of correct.
In a way, you could view it like that, yes. So
My question as well, so if the muscle is normally quiescent and the only time that the muscle essentially gets activated and you have a regeneration, and that goes well with generally, when you work out, there's pain and there's micro tears and that sort of stuff.
So that is a misconception, I guess would say. So let's go back to what I was saying before. The myofiber is often ignored in this kind of research, and the myofiber is very adaptive. So when you exercise, yes, you're right, you do tear your muscle a bit. It does cause injuries to your muscle, but recent research, I want to say 4 or 5 years ago, has shown that the muscle stem cells don't activate on that. The myofiber can fix itself in those minor injuries. So if you hurt yourself a little bit in exercise, it's mostly the myofiber that's just fixing itself because it's able to. And myofibers are also very responsive to the environment, to stimuli, and if you exercise and use them, they will just expand by themselves. So they are able to increase their protein content to expand and become larger. They don't actually need the muscle stem cells to help them expand. They can use them, and that's the case of injury. You do need the muscle stem cells in a catastrophic or large injury, but in small things like exercise, you don't actually need them.
And that makes sense, because if that was the case, then my follow-up question would be that doesn't really make sense, because then the more you work out, the more you're using your stem cell pool. And then as you're aging, well, it's just going to go down and down. Well, that's not going to be good.
That's also another interesting thought on it. So muscle stem cells, yes, they activate, they proliferate, and they fuse to the muscle, but there's always a reserve population. So when they expand, there's a couple cells that will return back to quiescence. So like I was saying before, every myofiber, every muscle has their own specific number of muscle stem cells they maintain. So I've seen in my work that, so the extensor digitorum longus, the EDL, the muscle I work on the most, it has on average in a young mouse about 5 muscle stem cells per myofiber. And then as you age, obviously you lose them. But if you injure the muscle and then they return to their quiescent state, you actually expand the number of muscle stem cells. So when we do an injury on the muscle, we give them 3 weeks. So that's the typical timeline to fully regenerate the muscle. And then if we isolate the EDL myofibers, we'll see that there's, instead of like the 5 per myofiber that you'll have in homeostasis, you'll have 20, 30 muscle stem cells on that myofiber. What's interesting on it is the muscle doesn't maintain that number. Like I said, the muscle has a specific number it wants. So after regeneration is done, I see from the molecular markers of those muscle stem cells that only about 5 or 6 of them have the true quiescence markers, and the remaining 20, 30 will eventually either fuse with the muscle or just die out from apoptosis.
Wow.
Yeah, so even though you're activating and using your muscle stem cells, they're always able to replenish themselves and return to their standard number. But you do lose them, yes, in aging. You gradually lose them. In disease contexts, you lose them.
It must be very interesting regulating environment. So how they figure out like how much of them should proliferate, how much of them should stop and go back to quiescence to maintain that.
Yeah, that's a big question in the field. I don't have a very good answer on it. I always theorize, and this is a complete theory, so I have no evidence on it, so take it with a grain of salt. But I was using, because we know from other people's work, if you watch like a myofiber and the muscle stem cells on it, even though muscle stem cells are quiescent, they do move around. Like they travel around the myofiber, they are able to move, they have philopodia, they can move around, and they bump into each other. And work from someone, I forget who now, and if he watches this, I'll be embarrassed because he'll know that I forgot his name. They showed that when they bump into each other, they trade a bit of their cytoplasm. So they refer to it as like a little kiss, like they just give a little kiss, they trade some of their cytoplasm, and then they move on. Moving back to your question on how ecology has helped me on this. So one of the things we know in entomology in ants is when ants will change their behavior based on how often they come into contact with other ants. So if you have a bunch of ants, they'll bump into each other a lot, and they'll change their movement patterns. So rather than moving in a meandering path, they will change to become more straight, and they'll go further and further away. So I always figured like maybe muscle stem cells do something similar. So the more frequently they bump into each other, the more they'll change their behavior. So if you're bumping into a muscle stem cells often, you might think, oh, we have a lot of muscle stem cells, I should just differentiate and fuse. And if you have fewer muscle stem cells, you'll be less likely to bump into each other and less likely to activate and differentiate. We also do know, however, that muscle stem cells are not a unique population. There are subclusters and different functional populations of muscle stem cells. The most common separation is in terms of quiescence. So quiescence, yes, like I said, it's that reduced metabolic state, no proliferation, but there's different levels of it. It's not an on-off switch. It's not you're quiescent or not quiescent. We have a population of muscle stem cells that are referred to as deep quiescent. So roughly 10% are more quiescent than the other muscle stem cells. So they will take longer to activate, longer to differentiate into myoblasts, and longer to enter that first cell cycle. And it's possible that that is your reserve population, the one that kind of stays on the back. Like say maybe you had a less serious injury, you don't need all your muscle stem cells to activate, they can just stay in their quiescent state and the others will take care of it.
But what differentiates between the ones that are deep quiescence and the ones that are like more closely to be activated?
So one of the main markers of it is the myogenic factors. So people may be familiar with PAX7, which is not a myogenic factor.
What are myogenic factors?
Sure. So the myogenic factors, or myogenic regulatory factors, or MRFs, they are the 4 transcription factors that are, I don't want to say essential because that's the wrong word, but very important for muscle stem cell formation, activity, and function. So they do go through let's start with adult stem cells because it gets more confusing with embryonic stem cells. But in adult muscle stem cells, you start in a temporal line. So you'll start with MIF5. So that's the first myogenic regulatory factor that activates. And that is present in your muscle stem cells, your more shallow quiescent muscle stem cells. Your deep quiescent do not have MIF5, but I'll circle back to that. And MIF5 helps in the activation and proliferation of these cells. MIF5 will also then induce the expression of the next myogenic factor, which is MyoD. MyoD will assist in, again, proliferation and expanding those myoblasts, but it will also then help in that shift of the myoblasts into differentiation. And MyoD will then help in the expression of myogenin. So myogenin is the next myogenic factor, and that one is necessary for the fusion of the cells together. So when myogenin is active, you have entered terminal differentiation, meaning you'll no longer proliferate. You have become basically a myocyte. And then it will help with the fusion of those myocytes either together to a new myofiber or to a pre-existing one. And then the last myogenic factor, MIF6, that one is expressed in the mature myofibers, and it helps maintain those myofibers and their function. So in terms of the quiescence, in the shallow quiescence, you'll have PAC7. So PAC7 is an essential transcription factor in adult muscle stem cells, even though it's not a myogenic factor. If you do not have PAC7, you do not have muscle stem cells. That's the simplest way to say it. And deep quiescent muscle stem cells only express PAC7. They do not express MIF5. So they're missing that first myogenic factor that helps the activation and proliferation, and therefore it probably takes longer to activate because of that.
And are the myogenic factors, are they just in the nucleus, or can they be found in the cytoplasm as well?
So these are transcription factors. So functionally, they have to be in the nucleus to work. However, like we say, your protein is made in the cytoplasm. So yes, at some point they are in the cytoplasm, but they do get shuttled to the nucleus.
So I'm asking because so your theory about like how they regulate, that's right, exactly, because if they're bumping, and there's different concentrations, so like maybe at a lower concentration it has I have no idea, I'm just at a lower concentration it has an activation signal, but when it gets high, it becomes the other one. So if they're bumping into a lot of new muscle stem cells that are going around that have these new myogenic factors, then the concentration is increasing. That might get them into the game.
It's an interesting thought, yeah. Again, I have no evidence on it, so I wouldn't be able to comment, but it's not just activating signals that you'll have for between quiescence activation. You do have the inhibiting signals or the pro-quiescent signals that are maintaining their quiescence. So like I said before, like notch 1, notch 3, they assist in the quiescence. But yeah, there's a lot we are not sure on, and I guess that's what all the research is important for.
It would be interesting to think, so how would we design an experiment on that, for example? Like you just...
Yeah, I'm not too sure on how you'd be able to do it.
Well, I guess the first step is obviously just go look at the literature, what's known on this, what's not known on this, what are the other co-founds, but it's just the communication of muscle stem cell I don't know, for some reason it's very beautiful. It's just going around the muscle.
Yes, I mean, a lot of work is being done on the concept of symmetric and asymmetric division. So just to define that real quick, so if you have one stem cell, it divides and it becomes 2 stem cells. That's viewed as symmetric division. Asymmetric division is when your muscle stem cell will divide and one of the daughter cells is more differentiated than the other.
Oh.
So some work mostly done in like Rudnicki's lab at the OHRI, they work a lot on asymmetric division to show, so when you have, let's say, a quiescent muscle stem cell, like let's take a deep quiescent one because that'll be easier for the analogy. It's only expressing PAC7, it divides once, and one of its daughter cells is PAC7 MIF5 plus, but the other daughter cell is PAC7 plus MIF5 minus. And that one goes back to quiescent, and that daughter cell that has the MIF5 will continue to expand to repair the muscle. So asymmetric division is known in the field, like it exists, it's a real thing. And other aspects of it is kind of what you were going for, is the cell, when it divides, it'll shuttle certain proteins to one daughter and certain other proteins to another daughter. So maybe more pro-activating proteins to one of the daughter cells and more pro-quiescent proteins to the other one.
No, it's very cool. Whatever scale you're looking at it, there's a lot of questions to be asked. It's like, okay, how does it regulate which proteins to like put where or not? Like when
Asymmetric division is not my specialty.
All right, and just looking ahead in terms of translating all of your research, what are some barriers in translating the work you do in the lab to more clinical sciences?
So there's a lot of issues with using muscle stem cell therapies in regenerative medicine. Let's start with, even though I don't work on it too much, but let's take muscular dystrophy since it's an easy example. So muscular dystrophy, like I said before, it's a disease where you have a mutation in the dystrophin gene. Your dystrophin, which is an important structural protein, it's kind of like your shock absorber of the muscle. So when you use your muscle, it kind of reduces the strain that's put upon your muscles myofibers. So when that protein is mutated, you don't have quite as solid myofibers. So when you do move around, use your muscles, your myofibers break very easily, and you need to go through regeneration, and you're stuck in this constant loop of regeneration. Your muscle stem cells get exhausted, and you're no longer able to make and regenerate your muscle. You lose your muscle mass. The reason I say it's a very simple example is we know exactly what causes the disease. It's a mutation in the dystrophin gene. Well, not a single mutation, like it can be anywhere in the dystrophin gene. There's more common sites of mutation. Therefore, it's an easy fix. You just fix the dystrophin gene. Very simple. But there are many problems with that. One is muscle stem cells only exist in a quiescent state as muscle stem cells in the body. So if you take them out to do some kind of gene therapy or to correct the mutation in the muscle stem cells, they're no longer muscle stem cells. They become myoblasts, and they will never return to muscle stem cells. You can force them to return, like obviously with chemicals and reprogramming, you can make them muscle stem cells again.
by mimicking the extracellular matrix and
Yeah, you can. That's actually some work that is being done in other labs on how stiffness and the extracellular matrix can help maintain muscle stem cell quiescence and muscle stem cell identity outside of the body. Some good and promising results in that, but that's just one of the problems. And it is a relatively simple fix. Like I said, you could use iPSCs, you could use, like I said, some of that research that's being done to maintain their quiescence. But then that brings us to the next problem, which is muscle stem cells are very bad at transplantation. There's a lot of problems in transplanting muscle stem cells into a new host or the same host if you're like extracting their muscle stem cells and then using them in the therapy. And that's because they don't engraft at all unless you prepare the muscle beforehand. So if I take a syringe full of millions of muscle stem cells injected in a muscle, you'll be lucky to get one that engrafts. So that means we do know how to get around this, and you need to prepare the muscle in beforehand, but the preparation is very severe. So the two ways that work reasonably well is either catastrophic injury. So how we do it in the lab and in other labs is you'll inject, for us it's cardiotoxin, so it's actually cobra venom. You inject it into the muscle, it melts the muscle basically, and then when you inject the muscle stem cells in it, they're able to incorporate into the niche and then become part of the muscle. The other method, also not pleasant, is high-dose radiation. So if you blast the muscle with a high dose of radiation, it kills off all of the host muscle stem cells, and then when you inject your muscle stem cells, they will be able to engraft into the muscle. But even then, you have really poor engraftment. So we've done this quite frequently in mice, like transplanting muscle stem cells into mice. And if you transplant 20,000 muscle stem cells, you might end up with 100 that have successfully engrafted into the muscle, which is still fine because technically you only need one muscle stem cell to regenerate the muscle. Technically, it's better to have a lot more, but you technically only need that. But then again, most patients would not appreciate either being bitten by a cobra or given really high doses of radiation.
Yep.
And naturally, the other problem is muscle stem cells don't travel between muscles. So if you would technically have to inject every muscle with muscle stem cells, and there's over 600 of them, and again, if you're doing like radiation, that's not really good for your body. You don't want to have severe injury on every muscle that you have. You could focus more on like the more important muscles, like say the diaphragm or some of the leg and arm muscles. But even then, that's still quite the challenge. The next challenge, this is why I chose the dystrophin one, because it highlights some of the limitations we have, is our main method of gene therapy is using viruses, essentially to incorporate the gene into the genome. You don't need to use viruses, but it's the main method and it's the easiest method. The problem is viruses have only a certain packaging capacity, like the size of the gene you can put in, and dystrophin is a huge gene. So the analogy I'll give is, imagine you have a box and you can fit 5 kilograms of something in that box. But dystrophin is 5 tons. So we're nowhere near being capable of having a delivery system using viruses to insert that dystrophin gene into the muscle stem cells to fix the gene. There are other methods. So people are looking at like exon skipping. So we know when you do a genetic screen on the dystrophy, you can see like which exon of the dystrophin gene has a mutation which causes the problem, and you can basically remove that exon, and most of the time the gene will just skip the exon and then just keep going. So you'll get like a truncated dystrophin that is still fairly functionally good, and that could be done with like CRISPR gene editing. Or you could do instead not do the stem cell therapy at all and just inject the dystrophin gene into the muscle directly. That would work, but it would only be a temporary fix, because once your muscle does break down, the muscle stem cells that are fixing it do not have that fixed dystrophin gene, and therefore you'd be going back to normal after a couple rounds of regeneration. So that would be, yeah, we have a lot of translating this. But that's, again, if you're doing like stem cell transplantation, like you could view regenerative medicine in just enhancing the regenerative capacity of the muscle stem cells of the host. So if you're not like in a genetic disease, like say sarcopenia, aging, muscle wasting, you could be looking at methods to either promote like the ECM remodeling, have better signaling molecules, promoting changing the niche environment essentially to promote the function of those muscle stem cells.
All right.
Is there, and I mean that I probably should ask another question before asking the other question. So in the example diseases that we talked about, there appears to be something wrong, and it's idiopathic in many cases of them. We don't exactly know what's the fundamental cause or anything, but there is something wrong with the endogenous muscle stem cells, what that we don't know about. So is there any work or any I don't know, some way to harness the endogenous muscle stem cells? I mean, if there's something genetic wrong, that they're not going to be able to fix it, but maybe if for example the dystrophy is caused by cancer cells, which was the other type, I forgot the name.
Cachexia. Yeah.
So maybe if we can, because I was thinking that, for example, we know that neuronal stem cells, they migrate in electrical fields. So like you can, you can direct them where to go by just putting them in an electrical field, and they follow that. And now muscle stem cells are not neuronal stem cells, but maybe, and so muscle does work with electrical fields, electrical pulses. Could something like that help in guiding the migration of endogenous muscle stem cells from some point to another, or it's just way off?
No, like you make a good point. So like in our lab, we do focus a lot on how the environment changes and how that affects the muscle function. And in aging, in cachexia, the muscle environment changes quite a lot. So I'll use some of the work. I was not the lead on it, but I was part of the project. Some of the work we've done on aging. So some work done by Dr. Felicia Lazur. She was a PhD when I was doing my PhD as well. She wanted to answer the question on, so you're aging, you have intrinsic changes in the cells, so DNA damage, accumulation of damaged proteins, mitochondrial dysfunction, but you also have extrinsic factors that are changing, changes in the circulatory factors, changes in the niche environment, changes in the ECM. So how can we decouple this from each other? So what is the main cause the main effect on muscle stem cell dysfunction and aging. Is it the intrinsic factors or is it the extrinsic factors? So her work, she isolated aged muscle stem cells and transplanted them into young hosts. So she was taking the old defective muscle stem cells and putting them into a young healthy host. And from her work, this is just a very brief summary, but from her work she found that the effect of whether it's intrinsic or extrinsic, it's basically a 50-50 split. So when you move these old muscle stem cells into a young niche, you're able to basically reprogram their transcriptome and bring them back to a younger state. So they more resemble younger muscle stem cells, but you can't do it for every gene. So it's about 50% of the genes that are changed in aging are able to go back to a younger state. But that also means that 50% are not responsive to the niche. So that's where we kind of get like, it must be a 50-50 split, that basically intrinsic factors can account for almost half of the defects and the extrinsic factors can account for almost half of the defects. But since the extrinsic factors can count for 50%, that's a targetable system. So you can target, like say, we think none of this is real, so example, like let's say we think, maybe EGF would be great for promoting muscle stem cell health. Let's give aged humans some EGF capsules to increase the amount of EGF in the body. That's something that could be done to promote the function of the muscle stem cells. Or we could go like, can we try to maybe remodel the niche in order to bring it more to a more youthful state? Because even if you ignore all the problems I was saying with like stem cell therapies, even if you do succeed in doing it, if the environment is no good, you might still end up with having poor muscle stem cells. The analogy for it is, imagine a house, an old house, it's full of mold and lead paint and it's damp and cold. The people inside it are going to get sick. The solution to that isn't adding a healthy person into the house, because that healthy person will get sick himself as well. The solution is trying to fix the house in order to make it so that the people inside can be healthy.
All right, now switching gears a bit, moving away from the research, now more into your teaching side of life. Which one do you prefer actually, research or teaching?
That's a good question. I like both of them because I find they do in a big way overlap. So teaching is, you're trying to convey information to people, help them learn, but in a way that's also research, because research is about conveying your findings in order to make an impact on the world itself. So if you're researching and not teaching anyone about it, then your research is meaningless. It wouldn't matter if I had discovered a cure for cancer if I don't tell anyone, because then nothing will come from that. So teaching and research, I find they do go well together. And I do enjoy both of them, but at this moment I probably enjoy research more, but who knows, that could change in the future.
There's a balance. It goes back and forth.
And what is your favorite part about teaching overall?
Well, I like to explain things, so that's always fun. But yeah, like I do have a good time teaching.
And you lay out information in a very structured way,
Exactly.
which is it's very nice when you like just put this like a structure. Like, let's go one by one. So first, let's talk about the structure itself, and then this one.
And also the analogies really help to knocking that down.
It ties it up together.
I think it's a good way to convey the information. You give the actual information, like here's what's going on, and then you try to bring a different context into it to see if it helps people grasp it in a better way.
Mm-hmm.
Yeah. And how about outside of teaching and research?
Yeah, I'm a pretty boring person. Mostly I'm at the lab, I do my research, but outside of that, I do play a bit of sports like curling in the winter, some tennis in the summer. I like cooking and baking, reading a lot. I have my video games, obviously.
Those are all exciting.
Exactly, you're being boring and having all these many hobbies.
We've had a lot of cooks among all the guests that we have here. And, always we have discussions about, we had Dr. Ardolino, and there was maybe 30 minutes we were talking about pizza recipes.
Yes.
No, but yeah, I mean, cooking and in a way research, they are very similar, because like lab experiments, here's a protocol, basically a recipe, like follow these, make it work, and cooking is quite similar in that aspect. So I guess there is some overlap in there.
And for cooking, it's much more like you get the reward much faster. Or if it's not working, you'll know much faster.
Yes, I do use cooking analogies a lot when I am explaining research, because I find they do overlap and go well together. And research can be difficult to explain, especially to people who are not in it, just how little you can see. Like, you're mostly, like, almost everything you're working on is a clear liquid. So with cooking, I can go with the answer, like, so if you don't know how to do an experiment, like, imagine you've never cooked before in your life, you have no idea what food even looks like, and I tell you, like, oh, add some eggs to the recipe. You might not realize you need to crack the egg and add it, you just toss it in, and then you have your shell in there. So in cooking, you see, well, that looks like a problem, but in research, you wouldn't. It's just like you've added a clear liquid to another clear liquid to another clear liquid, and then at the end result, you didn't get the reading you're expecting, but did something go wrong in that? You don't really know, so you have to kind of go back every step and go, did something go wrong here? How can I fix this?
And it's really hard. I mean, we've talked about all this, and then like there are little questions that are exciting, that are important to us. But then that one question that's like it's 5 words, but that can be a 4-year project.
Exactly.
Yes, sometimes the most simplest question we just don't have an answer for, or it's extremely challenging to get it to work.
Yeah.
And what is your favorite thing to cook?
Well, my mom's family is like Quebecois background, so a lot of my food is like more Quebecois, I guess. Maybe like a pâté chinois, I guess, or shepherd's pie. That's always fun to make.
Classic.
Yeah.
And if you weren't a scientist, what would you be?
Ooh, there's so many things I could have been, I guess. You never know where life is going to go, because I really liked like math and accounting as well, so could have been an accountant. I liked veterinarian sciences, so I could have been a veterinarian. Philosophy is a lot of fun, so who knows with that. Or I could have just been a farmer, like I grew up on a farm. I could have just stayed at home and
Lots of potential. Lots of
you
possibilities.
never know what's going to happen.
And now ending on your philosophical point. What is the
What's the
meaning of life?
That's good. So philosophically, I'd argue that there's no intrinsic meaning to life. Like, there's no meaning that you would be able to say, like, this is what life means, this is what has to happen. I'd say it's a more personal thing. So it's just the meaning of life is the meaning you give to it. So it's what you decide gives your life meaning, what you decide is important in your life. And I'd say it's a personal decision that everyone has to realize and come to on their own.
That's a beautiful thing.
Yes.
Yeah. All right. Well, thank you very much for being a guest on our podcast.
Thank
it
you
was
for
a
having me.
pleasure.
Thank you very much.
Yep.
Thank you.
Thank you.
Thank you for listening to this conversation with Dr. Blackburn. We discussed his path to muscle biology, his work on cancer cachexia, and more. If you're interested in his work, you can find a link to his publications in the description.
This is the Dendrite Podcast. To support it, please share this episode with your friends and anyone who might be interested. We hope you enjoyed this episode and see you next time.